Good morning, everybody. Welcome to the webinar. We're going to get started at about 8:03 as people trickle into the webinar. Good morning, everyone. We're going to get started in about a minute. Good morning, everybody. We're going to get started. Welcome to the Research Coordination NSF webinar for SARS-CoV-2 detection in wastewater. These webinars are focused on giving junior faculty and PhD students and postdocs the opportunity to talk about their research. And we're very thankful for our two speakers today to give us time out of their busy schedules. We have Cresten Mansfeldt, who's an assistant professor at Boulder, and we have Mitham Al-faliti, who I'm pretty sure I said his name wrong, and I apologize. I practiced beforehand, who is a PhD student at Howard. Cresten is going to go first today, and so I will turn over the webinar to Cresten right now.
Well, thank you very much, Ali, and also thank you for the invite to be able to speak today. And today I'll be talking about CUSew, which is the on-campus sewage monitoring network that we were operating over the past year at the University of Colorado Boulder. All of this was really inspired by the fact that PowerPoint decides to wake up today. It was just working. There we are. As everyone on this call is very well aware, is that RNA from SARS-CoV-2 is passed through fecal matter, and this enables us to actually establish these passive, semi-anonymous, and community signal monitoring networks on campus. And for the University of Colorado Boulder was no exception for experiencing the wave of SARS-CoV-2. What is being displayed here is the infection incidents detected on campus across the fall semester. So if you look at the date axis, it covers from when on-campus activity really resumed in August, all the way up until when students were asked to leave prior to Thanksgiving. And during the entire campaign, what is being represented on the y-axis there is actually all the different sites that we'll be introduced to momentarily. And then along the caseload is actually showing that the University of Colorado Boulder did experience a wave of infections after Labor Day, primarily due to increased social activities and intermixing of different social networks. And again, we saw a little bit of an uptick after Halloween as well. What we were able to do on campus to actually pin this infection data and understand it through the wastewater network itself is we established a daily cycle routine of collecting wastewater across campus. What is being depicted here is the cartoon of the overall idea of how we were actually operating on campus. We selected about 23 different sites of wastewater flow. We established pumps that would withdraw directly from the manholes themselves. During the morning time period, we would collect from each one of these pumps, bring it to the laboratory, and through a cycle, we would then get to a specific quantification of how much viral particles were present within that wastewater flow. And through temporal analysis, this would inform then the pandemic response office on what they would actually be able to do along the campus. They would pin this with other medical and diagnostic data to understand, do we actually have a full picture of the infection incidence on campus, and what social and actual public health mitigation activities can go undertaken to start to reduce the load and actually have those who are infected to actually seek medical services early in the profile. And then after this data was analyzed, we'd go back out, collect more data on a 24-hour cycle basis. Where we're actually operating on campus, what is being depicted on the map over there, anything that is highlighted in black is actually a University of Colorado Boulder owned building facility. And our pumps that were located, we had 23 pumps across 20 sites. And those are the red dots that are located along the sites. To be a little bit more economical and allow us to actually have a grid operating early, we actually established our own pump system. And for that, it only cost about $800 for the passive system that we had set up. And one advantage to this is it's, well, I've always called it kind of a stupid pump because it's just always operating. It doesn't do any back flushing, it doesn't do any time off, so it's just a continuous draw of the stream that is passing by that manhole. The selection of these sites was based on the ability to really subdivide the campus population. On average, each one of these pumps is representing about 400 students. And on campus during the fall semester and also into the spring, there's about 7,000 residents that were invited back to live on campus. One of the other considerations with this type of system was the safety of access to the manhole itself. One of the highlighting aspects that's up in the corner, you see a picture of Katie Reeves, the first PhD student on this project, and she really was a champion for deciding where to put these pumps and also how to design and deploy this entire system. When we brought the samples back to lab, it went through a pretty rapid concentration and analysis step where about 6 hours we could turn around the data. So what we'd end up doing is we spiked in a bovine coronavirus as an internal control. We just did a low-speed centrifugation to remove any of the large particles, and then we did an ultrafilter pipette to concentrate in the remaining particles and ended up with a column-based extraction. We achieved a concentration factor of about 20, and we were pretty happy with that sensitivity for the fact that we weren't monitoring at a wastewater treatment scale where we actually were only monitoring about 400 to 2,000 individuals per site that was being monitored. What we were actually seeing from the sample performance itself is when we were looking at the bovine coronavirus recovery. During the campaign in the fall, we had 3 different levels that we were actually looking at, where we started off at very low, moved to more intermediate, then really overcompensated with really high. But when we actually back calculated out what our efficiencies were, we saw overall, but there's about a 54% on average, but a pretty good standard deviation because of the fact that some sites were operating a little bit more variable than others. And then when we were looking at how confident we were in our quantitative comparison to our standard curve, we found that it was pretty stable over the entire run. And we were pretty happy with this type of profile that you see on the color-coded scatter plot over there. And that's just displaying the CT values from the qPCR method that we're using. We were doing a quadplex assay that was monitoring 2 targets for SARS-CoV-2, the envelope and the nucleic capsid, and then as well as our bovine coronavirus, and then also a sewage normalized control of the F+ bacteriophage. The majority of the variation that you see is actually resultant from the fact that we were using just a QuantStudio 3, so we were saturating all of our channels, and we didn't have a reference channel to actually cut out some of the noise. The actual data that we saw. So when we look at all the different sites that were anonymized on the y-axis over there, and as we turn the system on, the color coding goes from white to blue, with blue being much more intense in concentration. Gray is actually indicating that we're below our detection limit value. What you see is also there's some gaps in the data. The first major gap that's presented there is actually because of a early blizzard that went through the system. And then one of the other major gaps occurred because of a supply chain issue. But other than that, we were able to achieve on a daily basis the actual reporting of the concentration. And what we started to see is that there was this wave detected in the wastewater as well. The top bar chart that's up there is representative of the median envelope across all sites per day. And it's copies per liter, for it's actually normalized to the wastewater that we were collecting. And then on the other bar chart that's along the y-axis, that's actually now looking at what is the average incidence, so your envelope copies per liter per person over the entire stretch of the campaign run in the fall. And this is actually very reflective of the medical services data. But one of the other advantages that we had is we had a parallel data set as we were establishing this quadplex. We also had another data set that was being analyzed in this, was being displayed up here in this red heat map of the this is actually using the CDC assay, where it's using the RNA-sp, the nucleic capsid in the envelope, and this was run by the monitoring facility and allowed us to have a backup data set. One thing that is different about this red compared to the blue is the red was actually only one technical replicate. So this is actually indicative of if you're scrambling and you're trying to set up a pretty wide geographical and you're processing a lot of data, you can go back to a single technical replicate, but there is the inherent danger that you might lose signal or have more variability within what we're reporting back. Now, if we actually compare that blue heat map to our medical services data, we see that it is very reflective, as once we start actually detecting more incidents along campus, the wastewater signal goes up. And as we go into that quiet period, as we enter into October, the wastewater signal also goes down across a lot of sites, with a lot of sites actually going into the non-detect realm. And then after Halloween, we do see this increased incidence occur. This allowed us to actually respond at different stages. So if the virus is initially absent, we started to detect within the wastewater, this allowed us more of an early warning. And it actually allowed the Pandemic Response Office to deploy more resources to identify those individuals that would need to seek medical services. And then as we entered into more of the wave, we found the real strength of the wastewater data was to inform on the success of different intervention strategies. Were we accurately finding all those individuals that would need to seek isolation or seek medical services? And was it actually being reflected now within the sewage data itself? And then one thing is this data also allows us to see this post-peak, where as things start calming down and bringing it into a stable front, is now we can then return back up to the cycle and saying that the virus is initially absent. One thing that we did have on campus is we had a few isolation structures available. So the chart that's being presented has two different y-axes. One is actually showing the SARS-CoV-2 envelope per liter concentration on a log scale. That is specifically for this isolation structure. And then on the other axis, what's represented in the bar chart, is now the actual number of students that were asked to seek isolation across the entire campus. This is a slight unfair comparison. It will be swapped at one point because it's not all of those individuals within a single isolation structure, it's all overall. For the main isolation structure, you could imagine it's about half of what that bar chart is directly there. When we actually start now going back with some of the envelope, kind of a back of an envelope concentration here, where we can say that what is the SARS-CoV-2 per fecal matter? Because once we start understanding, it's like, all right, how much on average does an individual produce in wastewater? And within the residential structures, it's around 200 to 300 liters per day. And then what happens is we can figure out how much of that are we actually expecting to be fecal matter. And this is where we can get a rough estimate of about 10 to 9 copies per mil. One thing is that the valleys also indicate the ratio of this post-peak shedding to peak shedding within the isolation structure is shifting. The unique aspect about the isolation structure is all of the wastewater material moving out of it is contaminated with SARS-CoV-2. So when you start seeing shifts of the peaks and valleys, it's actually representative of those in isolation that are now moving through the viral progression and clearing the virus from their system. And so once we get into that valley period, it's actually indicative of that initial wave has now cleared. One of the other aspects that we had to have on campus was we were monitoring one of the administrative buildings. And this was associated with having to ensure that there wasn't a contamination signal above a whole bunch of other residential structures. This actually enabled us to also have a slight negative control built into our system. What you see in the charts here is actually the representation of the nucleic capsid and envelope concentrations. On the furthest left chart, in the center chart is actually our F+ bacteriophage genome copies per mil, and then the TSS per milligram per liter on the furthest on the right-hand side chart. What we saw here is primarily there wasn't contribution to the signal from this site. We had 2 days that there was actually a detection, but it wasn't actually a high level, and it didn't really align with when we actually saw more TSS moving through the system. There weren't actually administrators within the building, so it provided also kind of a handy negative control to be processing along with all of our other samples, in addition to our normal spike in negative control as well. But one thing to highlight about that F+ is it's another virus with another unique signal of an epidemiological strength. So over the course of our campaign, you see again the nucleic capsid and envelopes on the furthest left, TSS on the furthest right, and the center is the F+. You see that's actually going over 4 orders of magnitude. So we weren't happy of this signal to be used as a normalization strategy. So as we were shifting into the spring, we actually abandoned and retired the F+ bacteriophage and looked much more towards the PMMOV. When we moved into the spring and we're looking at the PMMOV, I just pulled off our last plate that we ran on May 1st, and this one actually is indicative that it's much more stable. So across all of our sites that we're monitoring, primarily we're seeing about 1 times 10 to the 7th levels are higher, with 2 of the sites being an exception. The administrative building that I was just discussing, where it's much more clear and there's not actually that much fecal contribution going through there, and a site that I label as fast flush, because essentially when you hear a toilet flush in the building, you can actually see it go through the pipe very quickly. So it's also moving through and there's not a lot of material that's able to be captured at that specific site. We also expanded to look at influenza norovirus. And one thing that we were happy about is that we did not detect any influenza in our samples as we're moving. And this is expected because of the mitigation strategies that we're putting in for SARS-CoV-2 have been very successful of also mitigating influenza spread across the United States. The 2020 map shows that there was a very high incidence of flu-like illnesses and also flu detections across the CDC's FluView network, whereas it's not seen in 2021, where the green is indicating that primarily the signal is not being detected and is clear across the United States. One thing I also had students look at is always just grab and look under a microscope. Primarily, we're seeing very negative, but for those in much more of the macro biology kind of scale, for epidemiology would recognize this almost immediately as this is a pinworm. So there is actual epidemiological information just by also scanning underneath a microscope. When we started comparing the fall to spring experience, it was a completely different experience that happened on campus. In fall, we had that surge quiet period with a little bit of resurgence. In the spring, we actually didn't really see a surge again. It was much more flatlined and kind of much more reflective of what we saw post-Halloween in the fall. Additionally, although we were seeing 1725 reported cases in the fall and there was 1812 on the dashboard for spring, only 592 of those cases were actually related to student activity on campus again, because of the fact that students were asked to move back into the residential structures after February 7th. And during that time period is also when we resumed sample collection along campus. And so this is what the wastewater data looks like for the spring campaign. It's a little bit more raw because we're still processing a lot of the information. But we do see that it's a lot quieter and there isn't as much of a surge within the data. You also see bigger gaps within some of the data frames. And this actually relates to some of the events that happened. And this was the major cold snap that also really impacted Texas, knocked us offline for about a week because we couldn't operate pumps safely during this time period. There was also a time period where they were predicting a historic level of snowfall where we were supposed to get up to 48 inches. So we preemptively shut down because we couldn't figure out how to go out into the field when there was 4 feet of snow. And then also there was time periods where there was very traumatic community events where for mental health reasons, we actually shut down for a weekend to allow the team to recover. One thing that we actually noticed is also we could see this almost in a correlation analysis from the data, is fall was really related to community spread and spring was really related to individual events. What I'm going to show you here is 2 correlograms. And so prior to going into that, blue just indicates there's a stronger positive correlation. Red indicates that there's a stronger negative correlation. And as you're getting lighter and closer to white, it's just mostly indicating that there's independence. There's no relationship between the 2 events. And what you're seeing along the diagonal is actually all the sites that we're monitoring. So in the fall, we actually saw a lot of correlation between the majority of our sites. But when we look at the spring, so just to back up and say how you read this is if you go from one site, you look at that box, and you look at the other site, they're correlating, that would indicate what the correlation is based off of the envelope concentration between those 2 sites. And within the spring, we actually saw almost no correlation between a lot of the sites. We got to much more of that they're pretty independent from each other with a little bit more of relationships between some sites that were monitoring the same structure, but also some sites that were very distant from each other. So then that might indicate some other epidemiological similarity between the 2, or else just random draw between them. One thing also our data set enabled was actually looking at some of the dilution. One of our trunk lines, we actually had 4 sites that were monitoring along. So we'll just give those the designation down below. And if you look at the envelope copies per liter times the population, you get a good normalization of what's happening. We had one event where there was a single case in A, and it was able to be propagated through the system. So that's 1 in 360, 730, 940, and 2040. But if we actually look at a different scenario, where we knew that there was still 1 in A, but we didn't expect that there would be an increase in this DBA structure, we were then predicting that there'd be about 3 to 4 cases within this region and 4 to 5 cases in the 2040, including those 3 to 4. So here we actually found 2 cases within 48 hours of this detection. And then there was also 8 cases found within 48 hours of this within the wider region as well. So this is showing that tracing along the sewer network is also an effective method. And establishing a baseline allows you to have this idea of how many cases you're anticipating. You still have to account for if it's a super spreader or what the expected viral load and where in the progression of the disease there are, but it gives you an idea of how many you should be trying to track down within these structures. Overall, the summary of what we were able to do in this, over the past year is we were on this daily cycle and really able to deliver data to get a more idea and anonymous method of what was happening within University of Colorado Boulder's wastewater. And in reflection, for $25 of muscle power, you can open up a stream of information. There's a lot of processing going on, but there is a very open access to wastewater within a society at the moment. And this is where we ended up defaulting to having semi-public information. If there were responsible public health information managers, as well as some of the wastewater treatment plants asking for information, we shared openly. But we refrained from posting this on a dashboard because we were operating at very exposed locations that are more indicative of sample origin. This allowed us to protect our sample sites for our operators, but then also for the pumps themselves. We only had 3 tampering events, 1 including a stolen one, and then it also protected our sample integrity from planting evidence or poisoning well events. And so with that, I would just like to acknowledge that there is a very large team that was involved in this entire procedure, with actually over 30 people involved directly in the lab itself. And with them, I couldn't have accomplished all of this. And so it's been quite a year, and I'll be happy to open this up to discussion.
Thank you so much. That was excellent. So there are a few questions in the Q&A. Just a reminder, if anyone has questions, you can post them there, and audience members can also upvote questions. This first one, I think you addressed some aspects of this throughout the presentation. So one was you referred to results as normalized copies per liter. If you could elaborate on the normalization method, I know you said you weren't using the bacteriophage, but what exactly, and I was also curious about how you estimated the population numbers within that when you were talking about through the sewer.
First, I guess I was going back to, we don't normalize to any of the other biomarkers that we're monitoring, so either to the bovine coronavirus or to the PMMOV. We use those as more internal checks to ensure that, all right, did we have any inhibition that we were seeing in the sample, and overall we weren't really seeing there. I guess in my mentality, I was talking about like normalizing to figuring out like how much fluid we actually were capturing to get back to the copies per liter aspect. So we didn't adjust it's kind of raw in that aspect where it's raw envelope copies per liter, but to get back there, we just had to adjust for the weights. And then on the second aspect, what was the second part of the question?
I guess I was curious about how you figured out populations within these samples. I would imagine there was exchange of people, and some of these were dormitories, but I guess I was just curious about that.
During the fall semester, we had both the census data, but then there was also daily Wi-Fi ping tracking. So we knew how active the actual buildings were. And then additionally, we had a very robust diagnostic and medical services. So when we were talking about the individual caseloads, we had a pretty good capture rate for anyone who actually was both seeking because of symptoms, but also they had a weekly screening of everyone in all of the buildings on campus.
Okay. And then the second part of this question, sorry that I didn't quite cover, was about the accuracy of spiked bovine coronavirus versus I think that should be SARS-CoV-2. If you have any thoughts about that discrepancy potentially.
Yeah, we never explored a head-to-head comparison of like actually purchasing a like an activated SARS-CoV-2 to try that as a comparison. And I think that's where it's much more useful for understanding is like, is there a technical issue with your data? Like, are you confident that your extraction procedure was very similar from run to run, rather than saying, all right, can we use it directly to normalize back on the SARS-CoV-2 concentration?
Yeah. Okay, so there's also a question about more details about your auto sampler. And then a follow-up to that is kind of how exactly you did this passive sampling. Exactly what do you mean by passive sampling when you talk about the sampling?
I would say passive because of the fact that we would really interact only with the stations once per day. And that was just a slight quirk in how I say it. It's not passive like the tampon method, but it's much more where it's pump driven. So it's a continuous draw, and each site is actually drawing up about 10 kilograms of wastewater per day. And then from that, we sample it on site and bring material back. So we have a 24-hour composite.
Got it. Okay, and I think you talked a little bit about how you're sharing results. So I'll, in the interest of time, I'll skip that question and ask the next one, which is if you plan to continue this in the fall semester.
This was actually discussion with the Pandemic Response Office, and we decided not to deploy this in the fall semester with the current trajectory of the virus. Mostly because a lot of the resources that this was really tied to are shifting as well, where we won't have access to isolation structure, and we won't have access to as robust medical or diagnostic services. So we actually said that the most economical resource was really putting it into vaccine deployment, because they mandated that anyone returning in the fall has to be vaccinated, as well as mental health services.
That makes sense. Okay, I guess if there are no other questions, which I don't see any in the chat, I guess we can move on, but I'm sure people can contact you to get more information about that sampler maybe. But thank you so much. That was an excellent presentation.
Well, thank you very much.
Mitham, you can go ahead and share your screen and get started.
All right. Thank you all for the invitation. Today I'm gonna talk about the wastewater monitoring of SARS-CoV-2 in the District of Columbia Metropolitan area. This work was done with Dr. Jesus Delgado Villa in the Department of Civil Environmental Engineering at Howard University. First, I would like to introduce our area of study. So we are monitoring a total of 7 wastewater treatment plants. 5 of these wastewater treatment plants are within the Washington Suburban Sanitation Commission, as we can see here in the map. We are also monitoring the Alex Renault wastewater treatment plant over here. And we're also monitoring the Blue Plains wastewater treatment plant, which is one of the largest in the country, as you can see here in the map. The flow ranges between 0.8 for our smallest wastewater treatment plant to around 370 million gallons per day for our largest wastewater treatment plant. The population that are served by these wastewater plants range between a little bit over 5,000 for our smallest one, and for our largest one, it serves more than 2 million people. So we had to have some considerations regarding where and how we are collecting samples from the wastewater plants. So first of all is the ease for the wastewater plant workers, the ease for them to get these samples. That's one consideration. Another consideration, we wanted different type of samples. Since the virus partitions with the solids, we needed solid samples, and we also needed influent type of samples. So with that taken into consideration, all of the plants were able to provide us with influent samples. These samples are either 24 grab samples or composites. Several of the plants were able to provide us with primary solids. Some of these plants don't have primary chlorifiers, so they weren't able to provide us with the primary solids. So then they provided us with the activated sludge, and another plant provided us with the gravity thickened sludge. So we collected these samples between mid-April and late December of last year. The frequency that we were collecting the samples were twice a week. These samples were stored at -80 Celsius. The reason behind that is because we had supply chain issues where we weren't able to get the kits. So we wanted consistency across all of our samples. So that's why we decided to store all of our samples at -80. When it came down to analyzing these samples, we all thawed these samples at room temperature overnight. So moving on into our methods. So before the samples were frozen at -80, the bovine coronavirus was added as an internal standard. So we are analyzing 2 types of samples, which are the solid samples and the liquid type of samples. For the liquid type of samples, we are using 2 types of methods. The first method being the Bromega kit, which I'll talk a little bit more about in the next slide. This concentrates the sample by 240 times, and then it gets quantified using real-time qPCR. The second method being HA filtration, where it concentrates the solid part of the sample, and then it gets through further analysis with the Qiagen kit, which then concentrates the sample by 96 times, and then it also gets quantified using real-time qPCR. For the solid samples, we are using the Qiagen RN Easy Power Soil Total RNA kit, and it concentrates the sample 6 times, and then it's also gets quantified by real-time qPCR. So I'm gonna explain a little bit about the Bromega kit. This kit is like an integration of both the concentration and the extraction steps. So you would save time on combining 2 of the steps together. Also, you start with 40 ml of your wastewater sample, and after you're done with all of your analysis, you would get 40 microliter of sample to analyze. So I want to just touch on about some of the steps that goes through the Bromega step. So first, the proteinase is added, and then the sample is incubated for 30 minutes. And then that sample gets centrifuged, and then we're only taking the liquid part of the sample, but not the solid. So then we use this MIDI column right here that gets inserted into a vacuum manifold. This manifold is attached to a vacuum pump to filter the samples. So we filter the sample through this column here, and then it captures the total nucleic acid. After several processes, this column is inserted into an elevator as shown here. This also, it's attached inside of it is the microcentrifuge. So we then get the solution by adding the nucleus-free water through this column, and then we would get around 1 milli of sample in the microcentrifuge, and then it goes through further analysis and extraction, and from that you will get your 40 microliter. For us, we're doing 6 samples each time. So that takes about 2.5 to 3 hours. So it saves time for us, and that's really critical for us to move on along with the analysis of our samples. So I'm going to present several results. The first result is a comparison between using the 2 types of methods, which are the Bromega kit and the HA filtration. And we want to look at the inhibition effects and the reproducibility of these results. So this first graph here shows the same sample done in duplicates for both the Bromega kit and the Qiagen kit. We are detecting the N1 and N2 copies, gene copies per liter, as shown here in the y-axis. For the Bromega kit, we saw that the results are reproducible, where we have almost similar gene copies per liter results. However, for the Qiagen kit, we have like one of the samples had a higher concentration, but the other one had a lower concentration. So the results weren't similar with that. So we wanted to look at the inhibition effects for these type of samples. So for the samples, we spiked them with standard SARS-CoV-2, and we detected the percent recovery for both of the methods. For the Bromega kit, we noticed that there are minimal inhibition effects. However, for the Qiagen kit, we noticed a lower percent recovery, which means that there are a lot higher inhibition effects using the Qiagen kit. To give this all in perspective, we know that the virus partitions, the envelope virus partitions with the solid. So we would expect higher concentrations with doing, in our case, the HA filtration with the Qiagen kit. we saw a higher concentration. However, that's not always the case. Where we saw one of the samples had really low concentration, and that's due to that inhibition effect, and one of them had a higher one. So when we compare that to the Bromega kit, we are for the Bromega kit, we only take the liquid part, and when we do the centrifuge, and when you take the liquid part, you would have lower concentrations of the virus, but it gave us more reproducible results and showed minimal inhibition when we compare it to the Chiagen kit. So with that, since we know that there might be some inhibitory effects with the solid, with the virus, we wanted to see the primary solid inhibition effects. So this graph here shows the same sample done in duplicates. And we also spiked the sample with standard SARS-CoV-2. And surprisingly, we saw low inhibition effects when we compare the percent recovery of the virus. And for this extraction, we are using the Chiagen RNeasy kit, and that kit is designed to minimize the inhibition effects, and it's designed for the primary solids. So that's why we see like low inhibition effects for the primary solids. So the second result I'm going to present is that the monitoring in the Al-faliti wastewater treatment plants. I'm only presenting here the influent samples. So this plant has flow rates typical between 30 to 60 million gallons per day. So it's kind of a medium-sized plant for across the plants that we are monitoring, and it serves about 300,000 people. So here we have a top and a bottom graph. The top graph shows the daily cases for the Al-faliti, and the bottom one shows the viral load. On the y-axis here, we have the daily cases, and on the y-axis here, we have the viral load. This load is normalized by multiplying the N1 and N2 copies by the flow rate going into the wastewater treatment plant. So these daily cases data, it was publicly available. And here we overlaid the sourceship over the cases by zip codes. So the dots here show the day-by-day cases, and the line here shows the 7-day moving average. So when we compare that to our viral load into the wastewater plant, so also here we have N1 and N2, and we see that N1 and N2 show similar trends across our samples. So when we compare this to the daily cases, we can see here by mid-May to June, we had like a high number of cases, and this corresponds the same way with the viral load into the wastewater plant. So then we have a decline in cases and a stabilization here. And then this corresponds also the same way with the viral load into the wastewater plant. And then by mid-November, we see a spike in cases. And this also translated into the viral load of on the wastewater, where we notice also an increase in the viral load to that wastewater treatment plant. So the last result I'm going to present is the monitoring in one of the WSSC wastewater treatment plants. I'm also presenting the influent samples here. The flow rates that are typical for this plant is between 0.8 to 1.5 million gallons per day. And it serves a population of a little bit over 5,000 people. This plant is the smallest plant that we are monitoring across all the treatment plants. So similar to the Al-faliti, the top graph shows the daily cases and the bottom graph shows the viral load. The daily cases also are the stewardship overlaid the zip codes. So the dots, they are daily cases, and the line is the 7-day moving average. So for here, for the bottom graph, for the viral load, the N1 and N2 genes show similar trends. However, when we try to compare this viral load to the daily cases, we don't see a real clear relationship. And this is due probably to the population served and the health data. For example, if we see here from June to October, in general, the cases don't really go over 5 cases, except in just some instances. So we don't really see the spikes in cases, and we're not going to be able to compare that weren't able to compare that to the viral load into the wastewater due to the size of the plant and the population served. So for our conclusions, we decided to continue using the Bromega kit, and this is due because of its availability, and we saved time on it. We are still continuing to use the HA filtration to archive our samples, and we're also still monitoring the inhibition effects within the Bromega kit. So when we saw the Alex-Renault relationship to the health data, we saw that the viral load exhibits the same trend with the health data. However, when we saw the WSSE plant, it did not exhibit the similar trend, and this is probably due to the health data and the size of the population served. So we saw that how the size of the plant can have an influence on the data. In that case, is that when we have fewer individuals that are infected, this obstructs our ability to establish the relationship between the viral loads going into the wastewater and the health data. And this also could be similar to the sewage samples, where if you have smaller number of people that are infected, how are we gonna be able to see higher the spikes within the cases, and also how that translates also the viral load into that sewage sample. So for our ongoing work, we are still continuing to extract and quantify both the influent and the primary solid samples from across all the different wastewater plants. We are comparing our results between all of we're going to compare all of results and the sample types also. And the reason behind that is that we would expect that with the different sample times, we would expect different inhibition effects. Also, we are working with our collaborators to model the differences between 4 different metro areas. Camille Makal, who is a postdoc at Rice University, is doing this work in parallel with us. And we're also collaborating with North Carolina State University and the University of Southern California. I would like to acknowledge our funders, the NSF, City of Houston Health Department, and the GHU CCTS Clinical and Translational Studies. Also, I would like to thank Dr. Hassef for her help and support with the project, our collaborators, and the utilities for providing the samples. And this is a photo of the Delgado lab. Thank you.
Great. Thank you so much, Mitham. That was a really excellent presentation. So as a reminder for anyone, if you have questions, feel free to add them to the Q&A box, and we'll work through as many as we can. And you can also upvote questions that you want to see answered as well. So the first question that we have here is about sharing your data. So I know that you mentioned in the beginning that some of those samples were collected and archived, so not necessarily processed immediately, particularly early in the pandemic, but can you share a little about how you're working to share the data with either treatment plants or the public health departments in your area, or possibly the general public? Do you have any sort of regular sharing with those people yet for your data?
We are still working on that. We still are analyzing our samples, so we're still like in the weeds of the work. And once we have like more data analyzed, we are gonna work definitely to try and share the results.
Great. Okay. Thank you. Next question is about your methods a bit. So you talked a lot about processing both the liquid and solid fractions with different methods and different sampling techniques. So do you have any thoughts on why the liquids and solids specifically would have high or low inhibition in your different samples or processing methods?
This comes to the partitioning of the virus. We would expect with it partitions with the solid. So with that, we would expect higher concentrations with the solids due to that partitioning of that.
Okay, yeah, but I think more specifically related to the inhibition you saw, not necessarily the SARS-CoV-2 concentrations. You know, I think it was that the QIAGEN kit had more inhibition than the Promega.
Yes.
Right?
Yeah.
So do you have a thought on why that was for like the different steps in the process? Why you might have seen more inhibition in that processing method?
Maybe it comes to the design of the QIAGEN kit. It might not eliminate these inhibition effects. So like for our this kit here, the RNeasy, it is designed more, as we saw with our data, it's more designed to lower those inhibition effects. Also, like for us, we only did like one, but we are still monitoring and seeing more how these inhibition effects within more samples that we are analyzing as well.
Yeah, great. Yeah, I know lots of, I'm not familiar with the exact steps in these kits, but they all have different, slight alterations in exactly the steps that you're doing. So the amount of material that you're cleaning out of your sample is going to vary substantially, I imagine. Next question, I had one come in on the chat, and I'm not sure if you know the answer to this, but do you have a sense for what the per sample cost is between the Promega kit and the QIAGEN kit?
Not right now, but I can get back to you for whoever asked the question.
Yeah, I know that's another, I know with supply chain, there's so many different factors that go into optimizing your method, both time and cost can be really important factors to consider. Yeah, that one came from Rolf Halden, so if you want to reach out with that info.
All right.
All right, so the next question here is for continuing your study. So I know that you're kind of retrospectively analyzing some samples right now. Are you planning to continue, or are you still collecting current samples, and are you planning to continue monitoring into, the future?
Right now, we are only considered with the samples we have. We have a lot of samples right now. So since we had the supply chain issues previously, we are now starting to generate data. And so once we have a sense of what we have at the moment, then we'll think of next steps as well.
Yeah, and I imagine it'll probably depend a bit on the interest in everybody's area is different as you show the data. To your collaborators, it'll be neat to see if they're interested in continuing long term.
Mhm.
So I have a question, one more question in the chat, and this is actually related to one I was just about to ask for myself, so that's perfect. This one's from Cresten. Have you looked at the effect of freeze thaw on your signal yet? I noticed that you froze some of your samples raw and that you included the BCO spike beforehand, so that might give you some information. Did you have any initial insights on how that process affected your samples?
Yeah, so so the one that where we added the B cove, the results weren't that good. Like the recovery was not higher than 50%. But right now we are trying to see the freeze thaw, where also like after we thaw it out, we are also trying to see those effects, but we didn't present it yet. But we are monitoring that as well.
And do you know if you saw yet any difference in the recovery of that B cove between your two different liquid sample processing methods, like doing the HA filtration after freezing versus the ProMega kit?
No, not yet.
Not yet. Okay.
Yeah.
Oh, and I see Heseth answered the cost question.
Okay.
So there's that information for everybody. Great. Well, I think if there's no more questions in the Q&A, I'm looking through my notes really quickly to see if I had any other ones. But I think we're getting close on time. So I think we'll close it there. And I'd like to say thank you to both of our presenters, Mitham and Cresten, for joining us today. Really great presentations all around. We're really happy to have everybody here, and we'll be having another webinar in another couple of weeks. So thank you again, and hope everybody has a great day.
Thank you all.